My name is Joshua Mugabi. I welcome you to Inheritance and Evolution. We on Learning Outcome C. Apply Mendelian principle to predict inheritance pattern and utilize mathematical models to analyze allele frequency and genotypic distribution within the population. The learning outcomes or learning activity for today, learners watch an animation or video on meiosis, focusing on homologous chromosome segregation and independent assortment, and relate this process to Mendel's law. Make a write-up and present their findings to the class. We are going to look at meiosis and Mendel's law. We are starting with homologous chromosome segregation during meiosis 1. Item 1. A biology teacher gives students chromosome strips made of colored paper representing a pair of homologous chromosomes. Blue strips equal to the paternal chromosomes, capital A. Red strips, this represents the maternal chromosome, small a. Students arrange this chromosome during meiosis 1, where homologous pairs separate. Task. Show how the homologous chromosome, capital A and small a, line up on the metaphase plate of meiosis 1. Demonstrate how segregation occurs during anaphase 1. Predict the allele content of the resulting daughter cells. Explain how this process supports Mendel's first law of segregation. Show how the homologous chromosome, capital A, small a, line up on the metaphase plate of meiosis 1. Homologous chromosome align as capital A on one side and capital small a on the other side on the metaphase plate. Demonstrate how segregation occurs during anaphase 1. During anaphase 1, A moves to one pole and small a moves to the opposite pole. Predict the allele content of the resulting daughter cells. Each daughter cell receives one cell, receives A, another cell receives small a. Explain how this process supports Mendel's first law of segregation. This demonstrates Mendel's first law because alleles of a gene separate during gamete formation, just as homologous chromosomes separate in meiosis 1. Let us now concentrate on crossing over and segregation. Item 2. During a laboratory simulation, students use beads on strings to represent chromosomes. Each chromosome has beads representing gene A and B. Teachers ask students to show crossing over before segregation. Task. Demonstrate crossing over between homologous chromosomes. After crossing over, Separate the homologous chromosomes. State how crossing over affects the variety of gametes. Relate this to genetic variation observed by Mendel. Demonstrate crossing over between homologous chromosomes. Crossing over results in chromosomes with new combinations of A and B alleles. After crossing over, separate the homologous chromosomes. After segregation, each gamete gets a recombinant or a non-recombinant chromosome. State how crossing over affects the variety of gametes. Crossing over increases the number of possible allele combinations, leading to a greater genetic diversity. Relate these to genetic variation observed by Mendel. Mendel's varied offspring phenotypes arose partly because crossing over creates new allele combinations before segregation. Let's now look at independent assortment during meiosis. Item 3. Students are given paper chromosomes. Pair 1, capital A, small a. Pair 2, capital B, small b. They are to simulate metaphase 1 alignment in 2 different ways to show independent assortment. Task: arrange the chromosome pairs in 2 different metaphase 1 orientations. Predict the gametes produced from each alignment. State the total number of gamete combinations possible. Explain how this supports Mendel's second law, independent assortment. Arrange the chromosome pairs in 2 different metaphase 1 orientations. Alignment. Orientation 1, capital A with capital B, small a with small b. Orientation 2, capital A with small b, small a with capital B. Predict the gamete produced for each alignment. Gametes. From orientation 1, we shall get gamete capital A, capital B, small a, small b. Orientation 2, capital A, small b, capital B. State the total number of gametes combinations possible. Total gamete types are 4. Capital A, capital B, capital A, small b, small a, capital B, small a, small b. Explain how this supports Mendel's second law of independent assortment. This demonstrates Mendel's second law because alleles of different genes assort independently during gamete formation, just as chromosome pair aligns independently in meiosis I. Let's now look at relating dihybrid crosses to meiosis. Item 4. A school garden has pea plants showing two traits: seed color, yellow, capital Y, and green, small y. Seed shape, round, capital R, or wrinkled, small r. A heterozygous plant, capital Y, small y, capital R, small r, form gametes through meiosis. Students must relate meiosis to Mendel's dihybrid results. Tasks. Using chromosome model strips to show the arrangement of capital Y, Y, and capital R, small r chromosome in metaphase 1. Show how independent assortment produces four gametes. Relate gamete formation to the 9:3:3:1 phenotypic ratio. State which meiotic process ensures this ratio is possible. Use chromosome model strips to show the arrangement of capital Y, small y, and capital R, small r chromosomes in metaphase 1. Capital Y, small y, and capital R, small r chromosomes align in two possible orientations. Show how independent assortment produces four gametes. Gametes formed are capital Y, capital R, capital Y, small r, small y, capital R, small y, small r. Relate gamete formation to the 9:3:3:1 phenotypic ratio. These gametes combine randomly during fertilization to give a classical 9:3:3:1 ratio. State which meiotic process ensures this ratio is possible. Independent assortment in metaphase 1 is responsible for the variety of gametes that produce Mendel's dihybrid ratio. Let's now look at human chromosome simulation in meiosis. Item 5. Students demonstrate human chromosome behavior using paper chromosomes for eye color, capital B, small b, and blood group allele, IA and I. Parents are capital B, small b, IA, I. Task. Show how homologous chromosomes segregate during meiosis 1. Show how independent assortment allow capital B, small b to sort separately from IA to I. Predict all possible gametes. Explain how these gametes explain variation among siblings. Show how homologous chromosomes segregate during meiosis. Homologous pair, capital B, small b, and IA and I separate during anaphase 1. Show how independent assortment allows capital B, small b to sort separately from IA and I. Independent assortment allows any version of capital B stroke small b to sort with either AI or I. Predict all possible gametes. The gametes, capital B, IA, capital B, I, small b, IA, small b, I. Explain how these gametes explain variation among offsprings. Different combination of gametes result in siblings with diverse traits, reflecting Mendel's laws. Let's now emphasize on errors in meiosis and violation of Mendel's laws. Item 6. A group of students studies non-disjuncture, failure of chromosome separation in meiosis leading to a condition such as Down syndrome. Task: using chromosome models, show what happens when homologous chromosomes fail to segregate in meiosis 1. Predict the number of chromosomes in the gametes. Explain why this outcome does not follow Mendel's law. State one real-life consequence of non-disjunction. Using chromosome models, show what happens when homologous chromosomes fail to segregate in meiosis 1. Both homologous chromosomes move to the same pole instead of separating. Predict the number of chromosomes in gametes. Gametes. Some have extra chromosome, n+1. Some have missing chromosomes, n-1. Explain why this outcome does not follow Mendel's laws. This violates Mendel's laws because alleles do not segregate or assort normally. State one real-life consequence of non-disjunction. Real-life consequence is trisomy 21 Down syndrome due to an extra chromosome 21. Let's now look at meiosis and the genetic ratios. Item 7. Students observe onion root tip slides and identify meiotic stages. They learn that arcuate segregation affects observable genetic ratios in the population. Task: identify cells in metaphase 1 and anaphase 1 from the slides. Explain how mistakes in segregation would alter Mendelian's ratio. Relate proper meiosis to predictable inheritance patterns. Identify cells in metaphase 1 and anaphase 1 from the slides. Metaphase 1, homologous chromosomes align at the equator. Anaphase 1, homologous chromosomes pull to opposite poles. Explain how mistakes in segregation would alter Mendelian ratios. Errors would cause unexpected ratios. For example, 2:1 instead of 3:1. Relate proper meiosis to predictable inheritance patterns. Proper meiosis ensures precise segregation and assortment, making Mendel's predictable ratio 3:1 and 9:3:3:1 possible. Thank you for participating in Inheritance and Evolution Learning Outcome C, applying Mendelian principle to predict inheritance patterns and utilize mathematical models to analyze allele frequencies and genotype distribution within populations. We have been looking at learning activity B, learner watch animation or a video on meiosis, focusing on homologous chromosome segregation and independent assortment, and relate this process to Mendel's laws. Make a write-up and present their findings to the class. If this has been very useful to you, visit me at Mugabi Joshua Biology on YouTube.